Thanks everybody for coming out to the public lecture this evening. And I'm going to talk about one aspect of our interdisciplinary work and mostly focus on visualization, but through different applications. As Jan mentioned, I direct what we call the SCI Institute. It's a Scientific Computing and Imaging Institute. SCI is the acronym, which we pronounce SCI in Utah, which, like around here, works. So we have many great ski resorts very close by. If you haven't been to Salt Lake City, we too have hosted the Winter Olympics in Salt Lake City. Before I get started with the topic of my talk this evening, I'd like to give a little bit of history and background. And one of the things I like to do when I give these types of talk is to say a little bit of something about the history of computer graphics and how much happened at the University of Utah, because a lot of people have no idea. This was back in 1965, where the person number one is David Evans, where he was brought from Berkeley to start a computer science program at the University of Utah. And the first person he hired was Ivan Sutherland. It was a very good choice. Ivan had created the first computer graphics interactive program called Sketchpad. Ivan would later receive a Turing Award for his work in computer graphics, also a Kyoto Prize as well. The third guy was Tom Stockham, and Tom was a signal processing person. He created the technology for the CD-ROM, so he created the CD-ROM. He also helped unerase parts of the Watergate tapes back during the Nixon era, which was either people thought that was great or not. But those three professors, with a few others, we have a call-in call already. Question. And a few others created this amazing environment where they pretty much created a lot of computer graphics. And you'll know some of these names. So John Warnock is number three. He got his PhD at University of Utah. He co-created PostScript, and then he co-founded Adobe and has been the president of Adobe since. Number five is Ed Catmull, got his PhD at Utah. He co-founded Pixar, and he's the president of Pixar and also Disney Animation. Number six is Alan Kay. He invented object-oriented languages, and also he co-invented the laptop computer. Won a Turing Award for his work. Number 10 is Nolan Bushnell. You might remember the game Pong, the first video game that he invented. He then founded Atari, the game company, and he sold that for a few $100 million in the 1980s. Nolan was back recently. He's written a book called Steve Jobs' Only Boss, and it was Wozniak and Jobs worked at Atari for Nolan before they founded Apple Computer, and Nolan tells the story about Steve Jobs coming to him and trying to get Nolan to invest in what would become Apple Computer. And he says that Jobs was always screwing off and not doing stuff, and he wasn't going to invest in that company, so he didn't think it was going to make it. And it's like, oops. Number 8 is Jim Clark. Jim Clark founded Silicon Graphics Incorporated, SGI, and then also Netscape, and then many others, Healthion, my CFO, etc. There's number 9, Jim Blinn. If you know your graphics, you'll know about Garreau shading. Number 11 is Henry Garreau, who got his PhD at Utah. And then Fong shading, another PhD at Utah, etc. So it's really kind of amazing we have this amazing history in visual computing. And the institute I had has a definite foot in one of the major research areas that we do is in visual computing. We're about 200 faculty, staff, and students, so there are 18 regular faculty and many research faculty and postdocs, et cetera, and about 80 PhD students that come from 8 different departments. So we're pretty much evenly divided into information and scientific visualization, into biological, medical, and geophysical image analysis, and into scientific and biomedical computing. So we have about 6 or so faculty in each of those areas, and then a number of postdocs and many students. So at the weekly seminars, we have about 60 to 70 people that will show up for that seminar, and we really have a critical mass in these different areas, which was one of my goals when we started moving from a research group to a center and then to an institute. And there's a lot of overlap in terms of people who work in visualization also work in image analysis and vice versa, and some in scientific computing, et cetera. These are some of the national centers that we direct, just to give you a sense of the diversity and the breadth that we have. I, along with Rob McLeod and Ross Whitaker and Dana Brooks, direct this NIH Center for Integrative Biomedical Computing. We're in our 16th year now and have been funded we will have funding for more than 20 years. Valerio Pascucci directs this Department of Energy Center for Extreme Data Management, Analysis, and Visualization. We have one of the NVIDIA GPU Centers of Excellence. Ross Whitaker directs the Center for Computational Earth Sciences. Martin Berzins directs a DoD Center for Computationally Designed Efficient Materials. And then we're also part of a number of other interdisciplinary large centers as well. Okay, let's move on to the topic of today. This is a simplified version of your brain. It was done by Tor Nordstranders, and basically it shows the partition amongst your brain of your senses and an approximate speed of those senses. About half of your brain, and all of our brains, are devoted to image processing. We are very visual creatures, and it's also the fastest of our senses. Next is touch, and then hearing and smell, and then taste. So taste, very important sense, but you probably wouldn't want to use it to go through a lot of data to try and find out things about it. So it would make some sense that since we have so much of our brains associated with sight and vision, and it's so fast, if we can figure out ways to take data, and that is often large scale and is often complicated, and find the right abstractions, find the right mappings of that data to our visual cortex, that might be a reasonable way to understand this large amount of data that we keep getting more and more of all the time. So let me share just a high-level view first, of a glimpse of some of these mappings for different applications. In the upper left is a large-scale, a visualization of a large-scale simulation of a spiral galaxy forming. In the upper middle here, we have a simulation of the stress tensor of a new polymer foam rubber that material scientists wanted to understand how the stress would propagate when they would compress it. On the right is one of the largest combustion simulations ever done by Jacqueline Chen at Sandia National Laboratory, who I believe was a plenary speaker here last year. And a visualization at one time instance of very large terabyte-sized data sets that show the complexity of the fuels mixing at that one time instant, but also understanding some of the underlying ignition and extinction events that are there. In the middle, we have a visualization using vector field visualization of the magnetic field of our sun. And in the bottom right, for example, is a visualization of trying to understand, being able to compare experimental fluid flow with computational models going from laminar flow all the way through the phase transitions until turbulent flow. And one of my colleagues spent 5 years, Valerio Pascucci, spent 5 years with fluid dynamicists trying to come up with a quantitative way to compare the experiments to the theory, and he was able to use topological analysis to find a way to count individual bubbles during phase transitions, which was something that they could relate to both experiment and to theory. It was a big science paper in Science Journal. We have up in the upper left here, beautiful work from my colleague Chuck Hansen on confocal microscopy visualization. So that's the forearm of a mouse paw where they've been able to stain to see the tendons and muscles and the nerves. In the middle, we have a visualization of a pool of jet fuel that's been set on fire using very large high-performance computing with full physics and chemistry simulation to look at the combustion. We have orthopedics over here on the right, here looking at hips. This is Jeff Weich's work on computational biomechanics and looking at hip dysplasia and arthritis. And more we're seeing data that is high dimensional but doesn't necessarily have a three-dimensional component. So in this middle left, we have a system that looks at epidemiology, so spread of disease, and it's an integration of GIS systems to look at these geography along with coming from multiple different data sources and databases from the different CDC and other health organizations throughout the different countries, and then the ability to do hypothesis testing to say, I have a hypothesis about this disease is spreading, how might that look and how would it work overall? In the bottom left, we have some comparative genomics and molecular biology. And I'll go into many of these in a little bit more depth, but these are the kinds of mappings that we're creating as visualization research scientists to take this data and try to come up with a way to better understand it and make discoveries from that data. Okay, some snapshots. One of the areas that we've made considerable research progress in at the SCI Institute has been volume rendering. And volume rendering is the technique of taking scalar data, often image data, but it can be any kind of data, and figuring out how to show the insides of that volume on a two-dimensional screen. My colleague Chuck Hansen, along with some of our graduate students, Gordon Kindelman and Joe Niss, created a generalized version of volume rendering, which is called multidimensional transfer functions. And what this did was instead of just having the volume rendering be dependent on one particular variable, which was usually, for example, an image intensity, now we could add in derivatives to look at different material properties and curvature and other parameter spaces. So we could now generalize this into many dimensions. And what it did was allowed for more specificity and understanding of what we could explore into that volume. So this is just a CT scan of a tooth. You go to the dentist, you get your x-ray and you look at it, and it probably doesn't look this colorful, but it could. What we've done here is taken just that intensity scalar value, but now we've extended this to a three-dimensional transfer function, and now we can pull in and understand more structure that's inside and basically better understand what's inside the volume. So this is one of my favorite examples of why this is important and why doing it for really large scale data makes a difference. So this is the National Institutes of Health Visible Male. He's the most famous dead guy in medical imaging. Many years ago, before death, so he was a convicted murderer in Texas, which is not a good place to be a convicted murderer. And before he was put to death, he donated his body to the National Institutes of Health. And after death, they imaged him at the highest resolution of the scanners at the time. And then at the very end, they froze the body and put a high-resolution color camera above, took photographs every millimeter, milled down, photograph, millimeter. And they made all that data available online. So he became the first gold standard for human anatomy. And the data from his body has been used literally in thousands of scientific studies to further the progress in imaging and biomedicine. So it's been a huge positive. The challenge was the data sizes. So it was the highest resolution that they could possibly do, and especially the color photography, that no one could visualize the entire data set at the full resolution when it was done. And so what everybody did was they downsampled. They took a certain number of pixels and summarized those to be one. And so they reduced the data set size. And what you got was a lot of images and visualizations that looked like this. So when we created our volume visualization system called ImageVis3D, it was able to visualize the entire data set interactively, and it had this multiple dimensional transfer functions. And what we found was he was covered with tattoos. So this is not a photograph. This is a volume visualization of the ink in the skin from the photography data. So our input is a set of two-dimensional photographs. And what we're doing is we were able to find the right transfer function that basically found all the instances of the ink in the skin. And if you think about how small scale a feature is of ink in the skin, it gives you a sense of that why for 10 or more years after the people had been working with this data, they had no idea it was covered with tattoos because they took the data and they immediately downsampled it and the small scale features went away. And so they were very surprised to see that you could see this American flag, even with a little red there. It says "Mom" across his chest here, and it gave us a sense of being careful with your data before you start to automatically downsample or do data reduction, dimension reduction, kind of all the usual things we do to filter our data before we start doing our quote-unquote real science on it, or real applications. And so this got us to thinking about, well, we should be careful about what we do with our data, especially if we have small-scale features. This is a version of ImageVis3D that runs on your phone and on your iPad as well. And what amazes me is that that is a visualization of a 256 cubed X-ray CT scan of a hand, and that's being fully rendered with lighting and shading on the GPU and CPU of the iPhone. And I'm old enough to remember, it wasn't that many years ago, that to do that would be like a $30,000 workstation, graphics workstation dedicated. And now this is in your phone. This is, in your pocket on your phone. This ability to do this kind of level of visualization on these devices has really started to change how we interpret our world, and in particular, the physicians have really taken hold of this to want to use this on a regular basis. And one of the areas that we've been able to work on with these physicians is in the treatment of Parkinson's disease using deep brain stimulation. And Parkinson's disease is the disease that causes motor tremor, to where there's drug treatments that can work, but at some point there's often the case of where the drug treatments are unable to work any longer, and so you basically aren't able to control the tremor and not able to hold a glass or hold the utensils to eat. And deep brain stimulation offers real hope for some of these patients because you can stimulate a particular area in the brain that can disrupt that circuit that's going around causing the tremor. So here is an example of a patient that's undergoing the deep brain stimulation surgery. So this is live brain surgery. You can see the shaking of his cup, and they're placing the electrodes in his brain and stimulating until they find the right area in his brain that is now going to disrupt that circuit. And you'll see that now he gets his normal movement back. It's almost like a miracle to see this, because these people cannot eat or drink, sometimes not even walk, and they get much of the control of their life back. So where do we come in? To note that this is not a cure. This is a treatment that there's a stimulator that's there, and it has a battery pack that's installed. And if you turn that stimulator off, you turn the battery off, the tremors come back. So where we come in is after the patient has had the surgery, they go and heal, and things move around a bit in there. And so they need to come back after they've healed to have that retuned and optimized. And the current way that it's done is pretty much a brute force search way, where they connect up the stimulator to the patient and their hands and trimmer. You can see how deep these electrodes can be, and along these electrodes are different places where they can stimulate. And traditionally now they start with electrode 1, stimulation site 1, voltage 1, watch. Okay, that didn't work, voltage 2, watch. All right, keep going, keep going. It sometimes takes 4 or 5 hours to be able to find the optimal set of stimulation sites and voltages. My colleague, Chris Butson, who's a computational bioengineer, he also is on the faculty of neurology and neurosurgery. He creates patient-specific models, large-scale finite element models, to stimulate all of those possible locations for where they could tune. And this is one time instance. You see the little yellow here. That's showing the voltage distribution that would come by for that particular stimulation site at that protocol. And what he does is he runs these on large-scale servers, does all the possibilities, sends a text message to the neurologist when all the simulations are done, and then they go with the patient. And they know specific areas physically where they're going to probably try and get the optimal case. And so within less than 5 minutes, they can usually find and optimize the location. So Chris has now just received a large grant from the National Institutes of Health to do a nationwide in the US study of this at Stanford and Cornell and University of Florida and other places that have deep brain stimulation. And this is a kind of I think, application that really shows the power of a combination of large-scale simulation coupled with visualization and interaction that you can carry, with you on your tablet. All right, a little more science one for the science types. I know there's a lot of fluid dynamics and combustion folks in the audience, and I thought they would appreciate this one. It's often the case in science that we have these very complicated results that we need to somehow untie and disambiguate and tease apart to understand. And in this particular example, it's a combustion simulation that has 10 different species that they're trying to understand how they all interact to create a more efficient fuel. Because we can't visualize effectively in 10-dimensional spaces, and pretty much outside of 3D is hard for us. Maybe there's some people in the room that could do 4D, maybe, but I challenge anybody in the room to be able to do 10-dimensional spatial visualizations in your head. What scientists do, is they take those 10 dimensions and they separate them out and they make 10 2D plots. So you have each of your species chemical species along the right here, and then you have time. And what they're doing is they'll watch all 10 of these plots at the same time, over time, as the simulation goes, and then they try in their brains to take pairs or groupings of those and then figure out what's going on. Well, 3 of my researchers, Ross Whitaker, Valerio Pascucci, and Sam Gerber, figured out a new abstraction, a new mapping where they could take all 10 of those dimensions and have a topologically equivalent summary of those in a 3-dimensional visualization. All right, and what they were able to do then is to see all 10 of these species at the same time in 3 dimensions. And one of the things that happened was that the researchers knew about this pure oxidizer and pure fuel level, but they didn't have any idea about this third whole other arm for local extinction. So they discovered some new science by visualizing these 10 dimensions which they have stared at for years and years in a new way. And here's just an example of what it looks like when you see all 10 on the left. And then we're going to move this, the 3-dimensional equivalent in the center of the screen. And then there's a ring that you'll see in the 3D visualization moving now. And that's the time. So if you watch where it moves across all the 2D ones at the same time, so they could see all the 2Ds. And now that's where it is in the 3D. And they were able to learn something new by this new visual abstraction and mapping. That's when you know you've done a good job. All right, let's take another large-scale data look. This is some amazing data that Mark Lavoie created. He somehow convinced the folks at the Uffizi Museum to give him full access to Michelangelo's David statue. He then used a high-resolution laser range-finding scanner and scanned the entire statue at sub-millimeter resolution. He then gave that out to the visualization research community and said, who could visualize all that data? And we could. And we're doing an interactive visualization of that data, and we're going to zoom in here so you can see the resolution of the individual triangles that make up that model. Not only could we visualize it interactively, but we could show different lighting schemes. So at different times of the day when the sun is at different levels, we could see what the statue looked like. So that was pretty cool, but a year or two ago, he resampled and scanned the entire statue at much, much higher resolution. So now it's almost 1 billion individual little triangles. And each of the frames that you're seeing, what you'll see here, are 2 gigapixels in size. Not megapixels like your phone, but gigapixels. So we're going to zoom into that same place in the eye you saw in the previous model. And at this resolution, you will not be able to see individual triangles. It's amazing data. You can see chips in the marble and sculptor's marks, etc. And these two guys, Chuck Hansen and Valerio Pascucci, where Chuck was able to do the parallel ray tracing that it took to make these images. And Valerio created a new data structure that's based on what are called space-filling curves that enabled him to interact with these 2 gigapixel images on not only supercomputers, but down on laptops and iPhones, etc. One of the things that we discovered when looking at this data that I don't think most people know is that at some part in the past, someone carved their initials in the back of one of his legs. And in the next sequence here, you'll be able to see when we turn them around that right there, there's M and N. Someone has their initials for life on the back of Michelangelo's leg. Pretty sure you can't get that close to the statue now, but it's just, it's amazing data. So we recently released an iPad and iPhone app called Gigapixel David that's free. And one of the things about the statue is that because it is such a high resolution, and if you had a high enough resolution 3D printer, you could print yourself out your own Michelangelo statue. The museum will not allow Mark to give out the data anymore, and we're only allowed to show a certain number of views. So Gigapixel David you can download that, and interactively at the resolution you just saw, you can view those 4 different views of Michelangelo's statue. So I think you should definitely do this. It's really great. All right. So I mentioned galaxy spiral arm formation. This is work that's done by Elena Dongnia, who was at Harvard University when she did this. She's recently moved to the University of Wisconsin. And it's a very, very large-scale simulation where she got multiple terabytes of data and had no way to visualize the data. She happened to be visiting the University of Utah, giving a talk, and heard about our group. She came up, asked me, and said, I have all this data. Do you think you could visualize it? And I said, I think we could. And so the next week, I got a FedEx package of 4 terabytes of Harvard disks. And we put them in. It took a while to read them in, but we were able to use a parallel ray tracing system to be able to visualize every single star, and there are billions and billions of stars here, with the correct velocity, color, temperature, etc. So she flew back out, and we worked for days and came up with several different visualizations that I think not only were great for the science that she was able to compare these with observation, but made some really beautiful visualizations. And speaking of beautiful astronomical visualizations, you should definitely go to Paul Woodward's talk on Friday to see a lot more really amazing astronomy visualizations. All right, now from the very, very large to the very, very small. This is the highest resolution set of images of the retina of the brain. This is part of the Connectome project, which is an international set of neuroscientists who are using mosaic electron microscopy to be able to try to come up with these high resolution images of small parts of the brain so that they can then track the individual neurons to come up with a 3D wiring diagram of the neural structure of our brains. So there are scientists all over the world who are taking different pieces and parts using this technology, and then the idea is that once they all do this, they'll come back together and coordinate and come up with a comprehensive wiring diagram of the brain. All right, so let me tell you how hard this is and how big this is. So if you see these stair steps over here, can you see my mouse over there? That's the size of one the highest resolution of electron microscope. So that's 4,096 pixels by 4,096 pixels. Remembering that your big, huge HD TV screen is only 2,000 by 1,000 pixels. So it fits in the little corner here. They have this amazing technology that takes and slices the thinnest part of the brain, images at the highest resolution of electron microscope, and then dumps the data. Slice, image, dump, slice, image, dump. Works 24 hours a day, 7 days a week. When it comes out, it does not look quite this nice. So there's registration that has to be done. My colleague Tolga Tazazin has a toolkit to be able to do that. When they get an image done, the resolution is 200,000 pixels by 200,000 pixels. The resolution is 2 nanometers per pixel. That's 2 times 10 to the minus 9th meters per pixel. That is how resolved it is. And then they do the next slice. Well, they can't image and do the same process when they go into the depth that they do across. And so we get this very, very high resolution imaging in the X and Y plane, but then in the Z plane, in the depth, it's much, much coarser. You can see how much it's changing, even though they're slicing, at the thinnest possible slice. When they get done with one of these little bitty volumes of data, it's about 16.5 terabytes of data. And that's when they call us up and they say, okay, we'd like to visualize our data. And the fastest way to get that data is we drive up to their lab, we pull their disk, and we take it down to our lab and we put it in. So the old SneakerNet work is still the fastest bandwidth. And we have the capability of doing this large-scale parallel volume rendering, like you saw with the Visible Human, to take that entire 16.5 terabyte data set and visualize it interactively. This is our Powerwall, which has 36 30-inch monitors put together that gives us 41 gigapixels of resolution. And because no one had ever seen this at this resolution, we had neuroscientists in the lab spending hours and hours looking at the data and finding new structures and writing science and nature papers about them and being all excited. But one of the things that they noted is after that, now they want more resolution, and the next version of this will be creating more than 300 terabyte data sets, and they want to do simulations on these. So it already was a high-performance visualization problem. Now it's going to become a high-performance computing problem and image processing and visualization problem in the future. So it's amazing. This is what they're after. This is one cell type in that data set after they've done the segmentation of that one cell type. And remembering how small this is, 2 nanometers per pixel, this is how complicated our brains are and how packed they are. And I think they have a considerable challenge of doing all the different cell types and putting this together from all the different little bitty small blocks throughout the brain to create the full 3D wire diagram. But it's certainly a grand challenge problem that'll be very interesting to see how it develops. All right, let's move to some higher dimensional data. This is Mariah Meyer. She is one of our young researchers at the SCI Institute. She thinks about informational spaces. So instead of 3D spaces and time, she thinks about high-dimensional data and over time. And she's been working a lot with biologists. She does things a bit different than many visualization scientists. Many visualization scientists, especially scientific visualization people, they often create a new algorithm or a system, and then it's kind of a one-size-fits-all. You munge your data into Visit or ParaView or VTK or one of the other systems, and then try to get it to work and visualize. So her technique is entirely different, where she goes into the laboratory, she reads the papers of the scientists, she interviews the scientists about their goals, she evaluates the current visualization tools that they have, finding out what they think works and what they don't think works very well, and then she goes off and prototypes a new visualization system that she thinks will solve that problem better. So instead of the one-size-fits-all, it's much more customized, and then she puts that into their hands, they try it out. She then does another version and another version, so you have this rapid prototyping, and over the months or even years of time, she will then create a system that solves that particular problem really, really well. So these are examples of 4 of her systems, and as you see, they don't look alike because they're solving 4 different biological problems. So here's one of my favorite examples. This is done with a comparative genomicist at the MIT and Harvard Broad Institute. Had a model for how to compare chromosomes from human, which are the outer ring, to chromosomes of other mammalian species, which are the inner rings. This is the first time they looked at the model, his model data, and these kind of broad red and green bands you see here are correct. All the other stuff is incorrect. So he immediately knew his model was wrong compared to the experimental data. So he went away and came up with an improved model. This is what it looked like. It's better, but he understood that he had a fundamental flaw in his underlying model. So he went away for several months and then came back, and here was the experimentally verified and published version of the model. Mariah asked him how long did he think it would have taken him to get to this point for this model without her visualization tool. And he said, quite honestly, I don't think I would have ever gotten there. I think it's a really great example of interdisciplinary collaboration working effectively, where neither of these people could have solved this problem by themselves, and it was only working together that they were able to solve this problem. One of the things I need to make clear of is the importance of interaction. So you just saw some snapshots there, and this is just a little video of the system in action. And so these biologists would spend hours, analyzing their data and looking and asking these hypotheses and questioning, and then coming up with trends and figuring out slowly what would make sense and making updates into their models. So I don't want to think that this just happened. They just looked at something and it automatically appeared to them what was true. It took a lot of interaction with the visualization system to be able to figure that out. But this is the kind of tools that Mariah is developing in order to do that. To give you another sense about how important interaction is, this is another new faculty member, Alex Lex, who joins us from Harvard this year. And he's created a large number of tools for information visualization. And let me just give you a sense of the kinds of things he's doing. This one is for set visualization. One of the things that most scientists understand is when you've thought about sets, you've often thought about Venn diagrams, and so when Jan was mentioning about the work we started with computational science and engineering, there's often a Venn diagram involved with that, and there's 3 circles of it, where you have a computer science part, and you have a math part, and you have an application part, and people often thought that the intersection of those 3 circles was the set of computational science and engineering. I actually think it's more complicated than that, but that's one of the usual things. One of the things that you find out is if you have lots of sets going into the thousands, tens of thousands, millions, even a few, more than 3, is hard to visualize using things like Venn diagrams. And so Alex has been spending ways of coming up with more generalized versions of looking at large-scale set visualization. And I have a little video about that.
Upset also visualizes elements and attributes. To demonstrate this, we have loaded a dataset about movies that contains movie genres, release dates, ratings, and popularity. The view here is aggregated by sets. Here we can see box plots of the attributes: release date, average ratings, and times watched. In the element view, we see a scatter plot of release date versus ratings for the whole dataset. It seems like ratings in the past were higher, but this is possibly a bias of the data collection. We can see that the number of movies in the sets is quite different. To get a better understanding of the size of the smaller bars, we can adjust the scale. Bars that break the scale wrap around themselves to keep them comparable as long as possible. Let's take a look at the documentaries. The box plots reveal that documentaries are rated higher on average than other genres. Documentaries also don't seem to mix much with other genres. Selecting their element reveals that most documentaries are recent productions. The war genre, which is also highly rated, contains a lot more classics. Looking at the intersections, we see that war movies are more often of multiple genres than documentaries. We hope you enjoyed our tour of Upset.
So one of the things, if you quickly look at that, and all that, the whole system and all of Alex's systems are open source and online, so it's fun to play around with that. You might not have picked up that horror films are the least highly rated. I'm going to skip over this and talk a little bit about some of my own research for the last couple of minutes on uncertainty visualization, which is, I think, an important but often overlooked part of the visualization process that we need to take really seriously and incorporate it into all of our visualizations. And I pose the question of when's the last time you've seen an error bar in a 3D visualization, including mine. And I use this as an example of why I think it's important. This is a reconstruction of a tumor that for pre-surgical planning consideration from MRI data. So on the right, you see the tumor from one MRI slice. The way we create that isosurface of the tumor on the left is by segmenting the tumor on the right, slice by slice, and then we recreate the surface. There are two things that I think are wrong with the representation, the visual representation. One is that there's no indication of uncertainty at all. Two, surfaces play a very important role in our perceptual system. So surfaces is how we navigate the world. And so we think when we see a surface that it's true. So we overemphasize the trueness of surfaces. So what we've done here is we've have a surface which our brains will automatically overemphasize the truthfulness of, and we have not dissuaded that at all. So we have no indication that there's any error. Let's check it out. Okay, so here's a piece of the brain. We're looking at it. An algorithm or a person could probably look at this pretty well and do a good segmentation. Where is the right edge of the tumor on this image? Well, if you take some algorithms that do segmentation from computer vision, these are the extent of those algorithms. So what I'd like to tell the surgeon is on the left in this area, I'm pretty confident of that segmentation and the associated visualization. On the right, I am less confident, and this is by how much. And this is the kind of information that I think we need to start including and have quantitative measures from uncertainty quantification to be able to back this up. So we've been working on some of these for different applications. This is an example for diffusion tensor imaging, which is a way of using magnetic resonance imaging to come up with a visualization of the structure of the white matter tracts within the brain. So this is a real brain. It's been opened up here, and you see the corpus callosum inside, and then the white matter tracts. And this is a reconstruction by Ron Kikinis, this beautiful version that was used for pre-surgical planning for tumors. And the tumors can push the white matter tracts or even envelop the white matter tracts. And the surgeons do not want to cut those white matter tracts because that's what carries the sensory information. Cutting those, depending on location, you could lose sight, you could lose hearing, you could lose movement in a part of your body. And so they want to be very careful to avoid cutting through those white matter tracts. But as you can see here, there's no indication of error. And we often look at these as these kind of streamlines. And let me show you the underlying data of what that looks like. So here is the raw data of the MRI diffusion tensor imaging. And what we're going to do is we're going to show you down at the voxel level the resolution of that data. And we've used the corpus callosum as an example to show you the kind of discrete level of what data we're actually using. We take that data and then we line them up, and then we estimate, interpolate a line that goes through those in order to get these beautiful images on the right. Now, no neurosurgeon is going to actually use that to do their surgery because they know it's wrong. And so how do we better make more accurate algorithms and also show the uncertainty? So along with one of my graduate students, we started looking at a lot of these algorithms, and in this case, we were looking at popular diffusion tensor imaging algorithms. And this is from two examples of exactly the same data, and just a quick glimpse, and you can see that they're different. So one is more accurate well, we don't know yet. They might be both not accurate, but they're different. And so we created a system which was able to interactively compare the algorithms. We also worked with our carbon fiber lab at Utah to create a phantom so we could put it into the diffusion tensor imaging systems and get a ground truth and compare the algorithms to ground truth. So we could either compare the algorithms to ground truth or compare them to each other. The blue would mean low areas of error, red is higher areas. And then we could look at quantitative information in terms of how much error and where that error is, and to better look at that. Before I say this, so there are many, many different applications from uncertainty visualization, and I don't have enough time. I have whole talks on just uncertainty visualization, but I think it's a really, really important area for the future. I'm going to end with just two little things that are brand new and are just coming out this week, literally. One is published in the IEEE Transactions on Visualization and Computer Graphics this week, and the other one is being presented at the Euro Visualization Conference that's in the Netherlands later this week. So the first one explores a collaborative work with Hanwei Shen, who's a professor at Ohio State University, and his brilliant graduate student, Xin Tong. And it's about trying to understand and visualize 3D vector fields of dense, complicated, large-scale fields. And the problem has always been you get these spaghettis, dense things, and you can never see anything except on the very outside. And so we created this lens system in which we can move away layers of the fields. You can do this in an interactive way for even very large fields, and then be able to strip away the complexity of the surrounding fields and yet see the fields nearby, so you get the kind of big picture view, and then you can drill down and look at some features in more of a detailed view. The other thing that is really cool is that we have ways of finding different features, for example, particular types of vortices, and you can say find all vortices, and you'll just watch these lenses open up throughout this 3D field and be able to see all these at the same time. And so any feature that you can characterize, you can do that with. We realized that the structure for this also had applications for diffusion tensor imaging, because it too is a set of these three-dimensional streamlines. And we found a way for pre-surgical planning, for example, to be able to go in and start stripping away these tensor fields to be able to better understand the locality and location of those diffusion tensor lines, the white matter tracks, with the tumor. So that was just a beginning, but we're working with some of the neurosurgeons and neurologists to come up with better versions of that. Mentioning the deep brain stimulation that I showed earlier, which I think shows so much promise for Parkinson's disease, and I think for many other applications too, there is uncertainty. There's both uncertainty in the stimulation protocols that are not well understood at this point, and there's also uncertainty in the simulations that are guiding some of the stimulations and certainly the optimization of those electrodes. So we're working, this is work with one of my postdocs, trying to create visualization systems where we can take many different simulations or from patient stimulations and visualize those and better understand the uncertainties that we're seeing. And we hope then to come up with better understandings of how to treat better, make the simulations better as well. Okay, so just put up our institute again. These are the many faculty, postdocs, students who did much of the work that I showed you. So I want to acknowledge their amazing talent and work. These are the productivity machines that I provide in the lab. We have 5 different espresso machines in case 4 break down. We also have a ping pong table, a foosball table, some exercise bikes, etc. What I didn't understand was how international ping pong was. It's like the international nerd sport, because we actually have, you can see it's on wheels, and we thought, we'll bring it out every once in a while and put it in a closet. It's always out. This is in the reception of the institute. You get off the elevator, and right across from this is our reception desk. And it was like, the people from China and India and Italy and Brazil and the US and Europe, they all played ping pong. So we have annual ping pong tournaments. I want to mention, since many of the people in the crowd are high-performance computing types, that Supercomputing 16, SC16, will be in Salt Lake City next November, and call specific attention to the visualization showcase, which I am co-chairing. And we would like to have your input and examples. So this year, in addition to the competition of having the top 6 videos for visualization, we're also going to accept a number of other videos that will showing great visualizations of science that will be playing throughout the conference. And so go and find out more, ask me, and with that, I'd like to thank you for your attention. Thanks very much.